Okay, thank you very much. And I'm so glad to be here. I hope that you guys will appreciate what I have to say. I have a lot of new material in this talk that I'm presenting for the first time here. So I'm excited about that. And let's just get started. So let me share my screen and put this up. And here we go. Okay, so the title of my talk is Glyphosate, Deuterium, Prions, and Neurodegeneration. And you can see from some of the pictures here that we're going to have fun with the topic here. A lot of new material, as I said, and you're going to be surprised, I believe. I want to start with a quote. Men occasionally stumble over the truth, but most of them pick themselves up and hurry off as if nothing had happened. And that's Winston Churchill. Here's an outline of my talk. And I just want to step back and talk about my personal history of this journey that I've taken into biology. I've gotten so excited about biology over the past maybe 12, 13 years. Starting with an interest in autism, wanting to figure out what was causing it, understanding early on in my studies that sulfate was a problem for autistic kids. There's something wrong with the sulfate system in those kids. I'm absolutely convinced that's true and that that's central to their disease. Around 2012 timeframe, I learned about glyphosate, and it fit so perfectly that I was quite convinced that glyphosate was the primary cause of the autism epidemic that we're seeing in this country. And again, I found that glyphosate would disrupt sulfate in a big way. So that made sense with the autism. Around 2015, I learned about what I suspect is a unique, insidious, cumulative toxic mechanism that glyphosate has. It's unique to itself and that would disrupt not only the sulfate system, but also the flavoproteins. And so I'll be talking about what the flavoproteins are and how they play a role in the deuterium problem. I didn't learn about deuterium until 2019, December. I was so excited when I heard about deuterium. I mean, I knew what it was, but I didn't realize the significance of it, how it can really wreck the mitochondria, disrupt the energy production systems in the cells, mess up the immune system. If you're not handling it correctly, the cells have a very sophisticated, clever mechanism for dealing with deuterium, and glyphosate derails it. This is what I'm discovering recently. Very, very excited about this new direction of my research, and in particular, prions and neurodegenerative disease is a big section in this talk that's brand new. This is the first time anybody's hearing about this from me, and I have I'm working out a theory for what's causing prion diseases and how the prion proteins work, and it's quite fascinating. If I'm right, I think it's revolutionary. So you'll probably find this section very interesting. And I'm going to go into SARS-CoV-2, and actually I'm going to show how the mechanism that goes on in the lungs in SARS-CoV-2 is actually, I believe, a way to help restore deuterium health, to protect the immune cells from deuterium through activities that happen in the lungs. And finally, healthy lifestyle, and then a summary. So here we go, introduction, the big picture. Deuterium is a natural rare isotope of hydrogen, but it has distinct physical properties. Exposure to high concentrations of D2O, which is deuterium instead of hydrogen in the water, is life-threatening. And so living organisms have developed sophisticated strategies involving specialized enzymes to deal with it. And you can see hydrogen has a proton and electron. It's the smallest atom. Deuterium has an extra neutron, which makes it twice as heavy. Mitochondria require low deuterium water to function properly. Very, very important. Choices of water and food consumption can influence deuterium exposure. Glyphosate disrupts the entire system that manages deuterium in the body, causing deuterium intoxication. And then I have this new hypothesis that the prion proteins actually are promoting recovery from deuterium overload. And then in severe reactions to COVID-19, a complex response ensues aiming at restoring mitochondrial health in the immune cells. So deuterium is called heavy hydrogen because it's twice as heavy. It has an extra neutron. It's present all over in the physical environment. You can't avoid it. It's a natural element that's present everywhere at 155 parts per million in water, which sounds small, but as you'll see, as it said in the next bullet, that is, in fact, 6 times the concentration of calcium in the blood. And we know that calcium that has high levels, is a major component, a mineral in the blood that plays an important role in health. So it has distinct physical and chemical properties compared to hydrogen, and in particular, it disrupts these ATPase pumps in the mitochondria that are the source of energy for the cell. And so deuterium management in the body involves, first of all, trapping deuterium in hydrogel, which is really, really interesting. And this gets into Pollack's work on gelled water. And And then the body invokes these specialized enzymes that are really good at picking hydrogen over deuterium. If a molecule that they're supposed to react with has deuterium instead of hydrogen, it won't react. It's really, really fascinating. So they can feed the mitochondria hydrogen rather than deuterium to keep them healthy. Now, sulfate, I'm not going to talk a lot about sulfate today, but people who know me know that that's a really big topic of mine. And I still maintain that it's absolutely important and essential, and that systemic sulfate deficiency, and in particular heparin sulfate deficiency in the body, is a major problem in our society due to the glyphosate, due in part to glyphosate's mischief. And it is a common underlying problem of many, many diseases that we face today that are going up. So the glycocalyx is this, the lining. You can see over here on the right, you have this glycocalyx picture here of these heparin sulfate proteoglycans that go all along the blood vessels, and they trap gelled water. This is all Jerry Pollack's work. They trapped gelled water. The gelled water actually pushes out protons and then it creates a battery. And that battery is an energy source for all the cells, all the tissues. The protons actually enter, and I talked about this in a TED talk many years ago. The protons, this is a hypothesis that I have, which I feel like it must be true because it makes so much sense. These from the gel, they enter into the cell and they travel along the cytoskeletal lines as like electrical wires, and they deliver protons to the mitochondria. And the cool thing is they deliver protons that are depleted in deuterium because the gelled water traps deuterium. And then sulfate is critical in order to maintain that gelled water in the glycocalyx, lining all the blood vessels, and also in the membranes of all the cells. So it's really, really important. Sulfate plays a critical role in maintaining that gelled water. And so these sulfated glycosaminoglycans become depleted in sulfate when they are chronically exposed to glyphosate. So this is a big, important slide. But now I'm not going to talk much about sulfate as we go forward. So now I want to talk about deuterium. And there's something called deuterium depleted water. You can buy it. I'm drinking it. I have some right here. And it is essential for the mitochondria to function properly. People whose water supply is naturally depleted in deuterium are healthier. Deuterium depletion maintains strong hydrogen bonds in the network, hydrogen bond networks in DNA, which keeps it stable. And so cancer is associated with excess deuterium in the mitochondria. So people can drink deuterium depleted water to help fight cancer. When they have cancer, deuterium depleted water becomes a useful medicine to take to delay the cancer's progression. And interestingly, a ketogenic diet is a deuterium depleted diet. Animal-based fats in particular are low in deuterium. Now, this is a fascinating paper. And look at this. This is from 1964. And they had mice, and they were able to produce this water that was deuterium instead of hydrogen, and they fed it to these mice in a 5% dextrose solution. And then they described what happened. And these mice, they said their appetites were voracious, they were very hungry, and constant savage attacks upon each other were continually in progress. They got very, very angry. Eventually, at varying intervals, the animals became hypoactive, lethargic, and dramatically weak. And then death occurred in all cases within 6 to 10 days. So incredibly, incredibly toxic. And I've been told that it tastes pretty much like water. So it's a very dangerous poison. But of course, it doesn't really exist in nature. It's very hard to make it even. Now, this is a study that showed for humans, very interesting. It's looking at statistics over a broad base. For all the different states in the country, they got information on the rates of depression and on the amount of deuterium in the drinking water. So you can actually, in the public drinking water, you can get both of those data. And you can see these charts, the depression up on the top, the deuterium on the bottom. And you can see that they line up quite well. And this shows a plot of the amount of deuterium versus the rate of depression across all these states, shown here with a very strong correlation coefficient, p-value of 0.0016. Very, very interesting that just the water that you drink and how much deuterium is in it can have an impact on depression. And now, interestingly, deuterium levels are variable in different fluids. And in fact, the Antarctic glacier water has much less deuterium than the water at the equator. So if you can live up in down in Antarctica, you can drink good water. And people who get their water from glaciers live longer, statistically. Deuterium is higher in rainwater at the equator than it is at the pole. And so that's because when the rain comes down, the water evaporates more when it's hot. And the water that evaporates is enriched in hydrogen rather than deuterium because the hydrogen is lighter. It's much easier for the hydrogen to leave the liquid phase. And that becomes interesting with respect to some interesting ways to get good water that I'll talk about later. So the body actually differentiates. It moves deuterium differently into different body fluids. And in particular, it gets excreted in the saliva. The blood has less than the saliva, and then it's actually actively suppressed in the breast milk. Breast milk is a low deuterium resource, food. So the salivary glands are trying to get rid of the body's deuterium. And breast milk, of course, to feed the infant a healthy, low deuterium diet. Now, I want to start with introduce glyphosate. And here's the big picture. It's the active ingredient in the herbicide Roundup, which is pervasive in our food supply. People use it in their yards. It's considered to be safe for humans, but there's increasing doubt that this is true. Glyphosate kills weeds by suppressing an enzyme in the shikimate pathway called EPSP synthase, and our gut bacteria use this enzyme to make essential aromatic amino acids for the host. And of course, the gut bacteria get harmed by glyphosate, and then this causes gut dysbiosis, which is a major underlying problem associated with many modern diseases. There are strong correlations between the rise in glyphosate usage in the United States and the rise in a number of different neurological, oncological, autoimmune, and metabolic diseases. And this, a paper by Nancy Swanson, shows all kinds of plots of correlations between glyphosate and diseases. Here's my new book. They already talked about it earlier. It was released by Chelsea Green in July 2021. I present extensive data on glyphosate's toxicity to animals and humans from the research literature. And I show how glyphosate disrupts sulfate homeostasis, which is the link to autism and to many other diseases, by the way. And I argue that glyphosate is insidiously, cumulatively toxic through its diabolical insertion into proteins by mistake in place of the coding amino acid glycine. And this is critical, and I will say more about this throughout this talk to explain exactly what I mean by that and the effects of it. And this explains why it has a very unusual toxic effect on the body to cause all kinds of different diseases. So my next topic is NADPH, which is essential to support the mitochondria. And here's again the big picture. This is NAD is nicotinamide adenine dinucleotide, and it's an essential cofactor in oxidation reduction reactions. And it exists in four forms, which it can have a hydrogen, it can have a phosphate. So you have NAD plus, NADH, NADP plus, and NADPH. And you can see no H, no P, has the H, but no P. Then you can have both the H and the P with the NADPH. And the enzymes that supply NAD plus and NADP plus with H are all specially designed to avoid deuterium. So the H here is a very valuable H, and the mitochondria take advantage of it by pouring that H into the intermembrane space to drive as a driving force behind the generation of ATP. So these mitochondria, they supply the energy to the cell in the form of ATP. They depend critically on a proton ATPase pump to make it. And the protons derived from NADH and NADPH are major sources of the protons to drive that pump. Deuterium gums up the pump like sugar in the gas tank, and glyphosate interferes with the supply of NADH and NADPH to the organism in multiple ways. So here's just a picture of mitochondria. And, there's many of them in the cells. Neurons and muscles have more than other cells. They have a membrane, they're separate units. They have this intermembrane space, is this pale blue. And then the blue-green inside is the matrix, the mitochondrial matrix. And that's where a lot of the activity takes place. Protons are pumped into this intermembrane space, and then they come back out, and they're the driving force that causes these ATPase pumps to produce the ATP as the proteins are coming back out of the intermembrane space. So here's a picture of the membrane. So you have the matrix in here and the intermembrane space here, and this is the cytoplasm up here. And so you can see these NADHs, and FADH2 is an intermediary that's also very, very important. These two are supplying those protons that are deuterium depleted and pumping them up into the intermembrane space. And then they pour back out here where you have this pump ready to make the ATP. And so you can see the hydrogens come in, and then they all pour back out naturally because of the gradient that's been created by the pumping in. Very, very critical part of mitochondria. This water that's produced from oxygen at the same time as the ATP is produced is deuterium depleted water, because it's using these protons that have been produced through this NADPH process. So here's a picture, again, cytoplasm out here, the intermembrane space, all these protons kind of gathering here at these ATPase pumps that are in these cristae that come in invaginations into the mitochondrial matrix. These pumps are going to get messed up by the deuterium. And that's going to cause, first of all, an inefficient production of ATP and reactive oxygen species. And that is a big deal because mitochondria get sick. When they have too many reactive oxygen species, they get sick. So this is a fascinating paper by this guy, Olgan, from 2007. There are around 15,000 ATPase pumps in a single mitochondria, and the numbers are mind-boggling. Proton force rotates these pumps at a rate of 1,000 cycles per second. Deuterons get in the way. This is this deuteron here. They resist letting go. They stall the pump. They produce a stutter, and that causes a release of these reactive oxygen species. And so they disrupt this proton-coupled electron transport, and this is a biophysical mechanism by which you can select protons over deuterons in the whole process of making all this run smoothly, and the deuterons will totally mess that up. So it causes decreased production of ATP and increased production of reactive oxygen species, damaging the pumps. Now, this is interesting here. This is a little bit of an aside here, but you have this enzyme, glucose-6-phosphate dehydrogenase. Many people have mutations in it. It's a troublesome enzyme that doesn't always work the way it should. It's an important enzyme for producing the H, NADPH, which has this safe H that's not D. So this is also, NADPH is the largest source of hydrogen during fatty acid synthesis in biology. And so when you buy something like chlorella, which is known to be sort of a healthy fat, you're buying a deuterium depleted nutrient because these animals, these microbes produce depleted fatty acids because of this G6PD, which is able to select hydrogen over deuterium to make this NADPH. And so these fats that are produced by these photosynthetic organisms, such as chlorella, are very healthy fats. So fatty acids are a preferred fuel for the mitochondria because they are deuterium depleted. This makes fat a healthy food choice. So now glyphosate, glycine, and flavoproteins, and here again is the big picture. Flavoproteins are an important class of proteins that transfer hydrogens to and from NADPH using flavins, FAD, FMN, as intermediaries. And so they have these, they bind to both the FAD and the NAD or NADP. And glyphosate's disruption of the shikimate pathway in gut microbes leads to a deficiency in tryptophan, which is a precursor. That's one of the aromatic amino acids that get disrupted. It's a precursor to NAD. So that's going to become deficient if the microbes can't make enough tryptophan. And tryptophan is also a precursor to serotonin, which is the feel-good hormone. And that may be the connection between glyphosate and depression and also between deuterium and depression, excess deuterium. You might link all of this to, in fact, the violent behavior may also be in those mice that were exposed to high levels of deuterium could have had the violent behavior because of serotonin deficiency linked to excess deuterium. My research strongly suggests that glyphosate suppresses enzymes that bind phosphate. So not just glycine substitution, but glycine substitution in specific places. And I wrote about that in my book. I wrote about the flavoproteins and I wrote about the sulfur system. Both of them are getting disrupted by glyphosate because these enzymes that do this all bind phosphate. NAD, and of course the extra P here, NADP has even more. NAD, FAD, and FMN all contain multiple phosphate anions, and the enzyme has to bind to those phosphates at sites where glycine is highly conserved. And so this gives them, these flavoproteins, a tremendous susceptibility to glyphosate's mischief through glycine substitution for those glycines in the protein. So here's my hypothesis. Glyphosate disrupts proteins that bind phosphate, and it kills plants by suppressing EPSP synthase. This enzyme binds phosphate in PEP, phosphoenolpyruvate, at a site where there's a highly conserved glycine molecule. Whoops, I have to go back there, sorry. Glyphosate is also a glycine molecule, but it has an extra methylphosphonate unit attached to it, which is this thing down here. And so attached to its nitrogen atom. And so the binding site for PEP has a highly conserved glycine, and there's room for the phosphate in the substrate that's reserved by the way the enzyme shapes. But glyphosate occupies that spot. So here's sodium phosphate and here's sodium methylphosphonate. You can see they're very, very similar. So glyphosate fits very nicely into the spot that was reserved for the phosphate of the substrate. And now the substrate can't fit. And this is what I think is happening in all of these enzymes that bind phosphate. And there are many, many enzymes that are crucial for biology that bind phosphate, and they're all going to get disrupted by glyphosate because of glyphosate getting into the protein and blocking the binding of phosphate. So the unit fits nicely in the pocket that's reserved for phosphate binding, blocking the phosphate binding. So this is just a pretty picture of these water molecules and these water wires, which are so cool. And it's a basis of one of the mechanisms by which these enzymes keep deuterium out. And the water molecules actually are these flavoproteins have this hydrophobic region that allows just a handful of water molecules, like 12, and they line up inside this cavity that's created by the protein. And they can pass hydrogen from one another. And so this is what's happening here. There's a hydrogen coming in, which makes this one have an extra hydrogen. This is a water molecule, but it has one extra hydrogen coming in. And then it kicks off one of its own. And each of them picks up a new one, kicks one off until it comes out over here. So down below, you now have 3 hydrogens over here and only 2 over here. It's moved through by passing hydrogens along through this water wire. And that results in, by the time you get over here, this hydrogen is almost guaranteed not to be deuterium, because these water molecules, even if they had deuterium to begin with, they wouldn't want to pass it. So it's a way of assuring that deuterium ends up on the other side. And the flavoproteins use this mechanism in their design of their enzyme, and they have to bind the flavin in order for this whole thing to work. So I want to talk a bit about succinate dehydrogenase, a very important enzyme in the mitochondria. And this enzyme actually plays a role in both oxidative phosphorylation, which is what makes the ATP, and in the citric acid cycle, which is what breaks down the glucose and all the nutrients to turn it into water and carbon dioxide. So this is a flavoprotein, and it extracts 2 protons from succinate, its substrate, and then it embeds them in the membrane in the form of ubiquinol. So this is coenzyme Q10, which many people take as a supplement, very important molecule in the mitochondria for keeping them working properly. And it picks up these 2 protons through this enzyme, and those protons are almost guaranteed not to be deuterons. And so when you have a genetic mutation, it's succinate dehydrogenase, you can run a risk of getting cancer. Many of the cancers, you can see this list of cancers: neuroblastoma, breast cancer, colon cancer, renal cancer, melanoma, uterine cancer, prostate cancer, endometrial cancer, bladder cancer, gastrointestinal stromal tumor. All these things are connected to deficiencies in succinate dehydrogenase. So here is a bit of biochemistry about succinate dehydrogenase. It has this particular motif. It's called a motif of a GXGXXG motif, which is a classic motif for flavoproteins. And in the case of succinate dehydrogenase, it's GAG, GGAG. So it's 4 glycines here. And this shows this is a way you can represent sort of statistically what the different versions of the enzyme might look like. And down here is where it's binding: FAD, NAD. These are all these molecules that are binding the phosphates. Over here is FAD. You can see it has these 2 phosphates right here. And those 2 phosphates, these P's here, is where it's binding, right around these 3 glycines, which are highly, highly conserved. So when you change these glycines into something else, the protein breaks. It doesn't work. It doesn't bind FAD. And you get into a lot of trouble. And of course, you can't deliver the deuterium-depleted protons to the mitochondria. And now the studies have shown that glyphosate suppresses succinate dehydrogenase, which is no surprise to me because it perfectly fits what I call my glyphosate susceptibility motif. And so this study on E. coli proteins found that it's significantly suppressed by 3 to 4-fold, 3 different components of this succinate dehydrogenase complex. And a study on rat liver mitochondria in isolation exposed to Roundup, they found that Roundup, in this exposure experiment, Roundup affected the succinate dehydrogenase, but glyphosate did not. And they suspected that was because the surfactants in Roundup were necessary to help the glyphosate to get transported across these mitochondrial membranes. And another paper that analyzed the mechanism of glyphosate suppression suggested that it disrupts binding of succinate dehydrogenase to FAD. And that's exactly what I would expect, given my glyphosate susceptibility motif theory. Here's another paper that shows glyphosate formulations induce apoptosis and necrosis, which is, different kinds of death in human umbilical, embryonic, and placental cells, 3 different cell types. And they showed that the key mechanism was through suppression of succinate dehydrogenase. And over here, you can see this is SD, is succinate dehydrogenase, glyphosate coming in and disrupting it, and therefore messing up the mitochondria in a big way. And here's another paper that shows that it suppresses NADPH reductase. So that's going to be a problem for getting back that H, that healthy H. This is the enzyme that supplies NADPH to the enzyme aromatase, which is an important enzyme that converts testosterone to estrogen. And this is aromatase deficiency is connected to autism. It's sort of like a super male brain. There's too much testosterone, not enough estrogen, because this enzyme is being disrupted by glyphosate. And this chart from this paper just shows that the reductase activity goes way down when you increase the concentration related to the concentration of glyphosate with two different pH conditions. And so this enzyme is not only for this enzyme, NADPH reductase is necessary not only for aromatase, but for all these different cytochrome P450 enzymes that are involved in many, many different reactions. So, and then in my book, I talk about other things, other enzymes that I found in the research literature. Glyphosate suppressed them. So we have the succinate dehydrogenase, the NADPH reductase, but also cytochrome P450 enzymes. All of them seem to be suppressed by glyphosate. NAD transhydrogenase, which is an interesting enzyme that transfers the hydrogen from NAD to NADP. And I mentioned G6PD before, glucose-6-phosphate dehydrogenase, and also Rubisco, which also binds phosphate, and it is the most common enzyme in the world. And plants use it in photosynthesis. So it's actually interesting to me that Rubisco is suppressed by glyphosate, because I suspect that could be hurting the carbon footprint, because you pulling carbon dioxide out of the air to make organic matter is what these plants do, and they need Rubisco to do that. And so I suspect glyphosate is playing a role in climate change for that reason. So now the lipid pathways and deuterium, my next section. And this is all pretty new material. I've only been talking about this over the past maybe 6 months or a year. The big picture here is that the synthesis of long-chain polyunsaturated fatty acids involves a series of reactions catalyzed by elongases and desaturases, these two classes of enzymes. And so when you make these long-chain fatty acids, the membrane is protected from hydrogen leakage, which is important, because that will lose the energy. You want to keep the hydrogens inside the membrane. The membrane lipids are released under stress conditions, and that's also very interesting. They're released and then they're modified. And there are 3 different ways in which the lipids can be processed through various pathways in the body, and they're all interesting. The first one is the cytochrome P450 enzyme-based production of endogenous cannabinoids. The second one is lipoxygenase, LOX-based production of leukotrienes. And the third one is cyclooxygenase, COX-based production of prostaglandins. So you get these 3 different kinds of products, all these different complicated molecules that come out of the fatty acids by adding oxygen to them in various ways and modifying them, taking away hydrogens, adding oxygens, changing those into interesting molecules, some of which have powerful signaling mechanisms, signaling power. And so these things can cause inflammation. They actually, through their signaling, they cause an upregulation of all these inflammatory cytokines that cause all this inflammatory damage that is associated with many diseases. And so most, if not all, of these pathways produce deuterium-depleted water, which I think might be the primary product. In other words, the purpose of doing these reactions with the fats is to produce deuterium-depleted water. And glyphosate likely disrupts many of these pathways. So I want to just give you a little bit of an education on fats. They're quite fun. You can see here we have the different examples here. This one at the top, stearic acid. These are all, this is just a carbon chain. And so this one's very plain. It just has all these carbons and they all have hydrogens attached, and that's a saturated fat. Unsaturated fats have one of these double bonds, like this thing here, these guys here. These double bonds, whenever you see one of those, it means it's lost 2 hydrogens. The carbons now are doubly hooked up to each other, and they've lost two hydrogens. Each one loses a hydrogen. So you're pulling 2 hydrogens out every time you make a double bond. And those hydrogens are going to be deuterium depleted because they're made from this NADPH, and they're going to be useful for making water. So the monounsaturated, you just have one of these. And in polyunsaturated, you have more than one. And then you can have a longer chain. So here we've added another piece to the chain. And then those become highly unsaturated, multiple of these, and they have to be at least 20 long to be called HUFA. And then we have some really long chains, like this is hexacosanoic acid, which is, so you can see all these H's here being shown. This is a long chain with no saturation. All the carbons are just one bond, and they will have all their hydrogens. But then you can have something like icosapentaenoic acid, which is EPA, DHA, EPA, highly unsaturated fatty acids that people take as supplements like fish oil. This one's got 1, 2, 3, 4, 5 double bonds, which means it's missing 10 hydrogens. And it's pretty long, not as long as the other one, 20 carbons, 5 double bonds. So this one, EPA, is one that is considered to be a healthy fat because it's highly unsaturated. And now here I have a picture of all these reactions that take place with these desaturases and elongases. So you see desaturase, elongase, desaturase. So they come and pull out more and more of the hydrogens out of the fat. And so this is showing that you start with omega-6, like linoleic acid, alpha linoleic acid, and then linolenic acid is omega-3. And so the omega-3 fats are considered healthier. They come down here and finally get this DHA, EPA. Those are those fats that are in the fish oil. And then these are like vegetable-based fats, omega-6s, and they come down, and these produce the prostaglandins and leukotrienes that are inflammatory agents. These are going to cause inflammation. But you see that you're using these desaturases and elongases to make these longer chains with less hydrogen. So here's a nice example of desaturases, of the fact that desaturases produce deuterium-depleted water. So here's a PUFA, and then it goes to a HUFA with an extra double bond. It needs iron. It takes 2 hydrogens and a water molecule and an oxygen molecule, and it ends up with 2 molecules of water. It gets 2 molecules of water. And for each molecule of HUFA that's produced, then you get 2 molecules of deuterium-depleted water. And FAD is required as a cofactor. You pull 2 hydrogens off of the FAD to make it 4 hydrogens here. You've got these 2 hydrogens plus the FAD to make the water. And so I think an important thing these are doing is making deuterium-depleted water. Now, this is a tough chemistry paper that I found quite fascinating, and it's talking about how you can use these fatty acids, growing these fatty acids, as a way to fix metabolism when your mitochondria are broken. And so what's happening is you have glycolysis, which is what anaerobic processing in the cytoplasm. That's the first step in the breaking down of glucose, and that ends up turning NAD plus into NADH. So it's populating NAD with these useful H's that are going to be deuterium depleted. And it feeds that NADH into the mitochondria. And then the mitochondria steal that H and put it into the intramembrane space. So when everything's going well, the glycolysis is producing NADH, and then the mitochondria are restoring NAD plus so that glycolysis can continue. So when the mitochondria are broken, that breaks down. Now you need another way to get back to NAD plus. And the way you can do that is by building fats. And so these D5D and D6D desaturases extend these fats out, make deuterium depleted water. Right here is a 2H2O. This is deuterium depleted water with these desaturases, pulling out FADH2, these 2 hydrogens, as well as 2 hydrogens from the fat to make the water from the oxygen. And that is going to help the mitochondria to restore their healthy water so that they can get going again. So this happens when the mitochondria are unable to restore NADH. These guys facilitate the regeneration of the NAD plus so that glycolysis can keep going in the cytoplasm. So you end up with a cancer cell that's doing lots and lots of glycolysis and not doing so well with its mitochondria, producing most of its energy from the glycolysis and relying on the building of these long chain desaturated fatty acids to replenish NADH and to produce deuterium depleted water. It's quite interesting and complicated, I'm sorry to say. So when the electron transport chain is sick, too much deuterium, the cell naturally reverts to creating fats that are more highly unsaturated by extracting the hydrogen in the fats to make DDW and repair the mitochondria. And I think glyphosate is interfering with that because these are fibrin binding proteins. This is a cytochrome P450 enzyme. And this enzyme actually takes arachidonic acid and turns it into an endogenous cannabinoid. And so I think this is very, very interesting because I think this is being disrupted by glyphosate, which is why people are finding cannabinoids to be such a, like THC, to be such a useful supplement to take to help with cope with pain and things like that. If you had a working system with your cytochrome P450 enzymes, you would make your own endogenous cannabinoid and you wouldn't need the marijuana. And so these CYP enzymes are being suppressed by glyphosate. And that is likely because of this reductase problem, that is the enzyme that maintains the NADPH. The H on the NADPH is maintained by this enzyme that glyphosate disrupts. And so interestingly, when this CYP enzyme activity is suppressed, then this arachidonic acid gets redirected into the lipoxygenase pathway, the LOX pathway, which is going to generate these molecules that are inflammatory, that inflammatory signaling molecules that are going to cause inflammation. So there's your DDW right there coming out of this reaction. So inflammation, cancer, and oxidative lipoxygenase activity are intimately linked. This is a very interesting paper that talks about lipoxygenase, which converts arachidonic acid into these leukotrienes, which are pro-inflammatory. And ultimately, when you keep going, you get to lipoxins, which are anti-inflammatory. So this whole process is going through making deuterium depleted water, getting things healthy again, and then finally turning off the inflammation. The leukotrienes activate this NF-kappa B pathway and the TNF alpha expression, which are these inflammatory signals causing inflammation in the joint, causing joint pain, all these problems. And also in the brain, of course. So 15-LOX is one of these lipoxygenases that's highly expressed in white blood cells. And when those white blood cells overexpress it, you end up with many chronic diseases. It's associated with many chronic diseases like asthma, atherosclerosis, rheumatoid arthritis, inflammatory bowel disease, dermatitis, and cancer. All these things are connected to overexpression of lipoxygenase in the white blood cells, probably because the CYP enzymes aren't working. And this one in particular, LTP4, is a potential stimulator for cancer cell growth. And it also plays a role in reactive oxygen species in response to low oxygen. Lipoxygenase, interestingly enough, is a really fascinating enzyme. It has a fantastic ability to select hydrogen over deuterium, much better than the flavoproteins even. It's a really an ancient enzyme, extremely clever, does not bind, doesn't have the glyphosate susceptibility motif, which is really, really interesting. So I think it may be an enzyme that actually is protected from glyphosate's toxicity, and therefore tends to be the one that plays an important role, much more important than it should, because the other enzymes are broken by glyphosate. But it causes this inflammatory response. And so endocannabinoids have been found to be useful for many, many treatments. And this is a paper about cancer treatment. And there is a cannabinoid receptor, and this is an agonist to that, and it inhibits cancer cell proliferation through various receptor-mediated mechanisms. Now, if you had a human cannabinoid, it would do the same thing, but you don't because the cytochrome P50 enzymes are messed up. And so they showed that it slowed the growth of this Lewis lung adenocarcinoma in culture and in a mouse model after oral administration. It found that many types of cancer cells upregulate the cannabinoid receptor. The cannabinoids suppress metastasis and angiogenesis. And then these breast cancer cells that have this feature, cannabinoids arrested cancer cell proliferation in vitro and in vivo by inhibiting these signaling mechanisms. And it locks the cell cycle in these early phases so that the cell doesn't continue to reproduce, which is how the cancer grows. And so they've also seen anti-tumor properties in pancreatic cancer. So just in general, cannabinoids protect from cancer or treat cancer, and endocannabinoids would do the same if the body was able to make them properly. So now we're at the prions and the neurodegenerative disease. Brand new slides here, and this is new research that I've been doing furiously over the past few months. I've been fascinated with prion proteins for a long, long time. I really want to understand what they're doing. They're very puzzling to the people who research them. So just to tell you what they are, first of all, these are prion proteins. That is, the prion protein is the one that causes Creutzfeldt-Jakob disease, CJD, which is a human form of mad cow disease. All of these diseases are debilitating neurological diseases. Then you have these other proteins that are called prion-like because they have similar properties to the prion protein. All of them have a problem with misfolding under certain conditions. And when they misfold, things go south. The cell's not happy. And so amyloid beta is the one that's associated with Alzheimer's. Alpha-synuclein is associated with Parkinson's disease. These two are associated with ALS, Lou Gehrig's disease. And then you have the prion protein, which, as I said, is CJD. So all these diseases are horrible. They're going up in prevalence over time, and they're associated with misfolding of these proteins. So researchers haven't even been able to figure out exactly what is the toxic effect of the misfolding. They don't even know whether it's those fibrils that accumulate in the plaque, or the Lewy bodies for the Parkinson's disease, this Alzheimer's plaque. Is it those insoluble fibrils, or is it some soluble oligomer? Because these proteins buddy up with each other to make multiple copies stuck together in these soluble forms of oligomers. Is that the toxic effect? Or perhaps even it might be a loss of function, because when they misfold, they can no longer do what they used to do when they weren't misfolded. And they haven't even been able to figure out what it is they actually do, even though they know that if you have a mutation in one of these proteins, chances are you won't be born, embryologically lethal. So it's so fascinating that they have a very important role to play, and we don't know what it is. So here's the big picture. And this is the question I asked: do the prion-like proteins play a role in maintaining low deuterium in the cytoplasm? This is what I'm thinking is going on. And how this is happening is because I think these proteins are trapping deuterium inside water inaccessible precipitates. And so what I think is happening is that these proteins are able to pick up deuterium. And when they pick up enough deuterium, it causes them to misfold. And when they misfold, they collapse into this form that the water can no longer get to. And therefore, they can stop exchanging deuterium with hydrogen. And they hold on to that deuterium as a piece of trash, really, to keep it out of the main cytoplasm. So my bold claims are that misfolded proteins in Alzheimer's plaque or Lewy bodies are a sign of impaired deuterium homeostasis. Prion diseases begin with stressed immune cells in the gut and spleen. Cells with impaired ability to clear misfolded proteins release them inside exosomes and deliver them to other cells, propagating the disease process. Impaired sulfate homeostasis plays a significant role in the pathology. So then I have several papers that I've been reading to try to see if I can fit this theory into those papers. It's really quite fascinating. And this one says structural organization of brain-derived mammalian prions as probed by hydrogen exchange. So this is a very interesting paper to me because what they did was they figured the these proteins, when they're not misfolded, they're kind of hanging out in the cytoplasm, all sprawled out, and they have complete accessibility of their backbone to deuterium in the water. So they actually exchange the hydrogen that's attached to the protein with deuterium back and forth. Sometimes they get another hydrogen molecule, sometimes they get a deuterium. It's just back and forth, exchanging with the water freely. And people have used this as a way to find out, to understand better what's going on with the misfolding. And so this experiment actually looked at hydrogen deuterium exchange, and it used this method to look at the backbone amide hydrogens and within the unstructured regions, which is where they compared to when it was the slow exchange. So very slow exchange of those involved in the H bonded structures of the beta sheets and alpha helices. So this down here is beta sheets. This is the form that's the misfolded form. That natural form is just hanging out in the cytoplasm, picking up deuterium molecules. Once it gets too many, I believe, it causes it to refold into this tight structure that no longer communicates with the water. So it has grabbed the deuterium and held it into this plaque, keeping it away from the cytoplasm, reducing the deuterium level in the cytoplasm. So the prion protein gathers deuterium from the medium and traps it within beta sheets when there's too much. That's my basic theory here that I'm playing around with. So here's just some more papers, protein misfolding. A nice picture here of a whole protein folding is an incredibly complicated process. We don't understand how it works. And we know that the protein has to fold in a certain way to do its job. And so the proteins are synthesized in the ER, the endoplasmic reticulum. They go out into the cytoplasm. They can be picked up by chaperones, and then they can eventually be cleared. They do their job, whatever it is, and then they get cleared by ubiquitination. They go into this proteasomal degradation here, or they can come into these lysosomes and get broken down there. They can get broken apart into individual amino acids and then rebuilt into new proteins. So it's a constant process of building proteins, using them, clearing them in a cycle, but sometimes they get folded into some way. This is a native folding here. Now it can do its job, or they can get into this agrosome. This is when they've misfolded and they just accumulate in this plaque or in a Lewy body, this kind of thing. So this is just sort of the process of the whole protein synthesis and degradation, which goes on all the time in the cell. And this is just another paper that shows you the lysosome. Lysosome plays a critical role in removing misfolded proteins or in clearing proteins that no longer serve a role. And you can pull them in from the outside into the plasma membrane and carry them in through endocytosis and push them into the lysosome. Or you can actually grab some that are inside the cytoplasm into this macroautophagy or microautophagy, all these different mechanisms to sweep up these proteins, pull them into the lysosome. Lysosome gets very acidic. It breaks it apart into individual amino acids, and you're done with removal. But when you get, and then I guess there's some stuff going on over here too. So this is just the lysosome, macroautophagy, microautophagy, endocytosis, proteasome, ubiquitin-mediated protein degradation. All these things are happening all the time in the cell, in a healthy cell. Amyloid proteotoxicity initiates an inflammatory response blocked by cannabinoids. So this gets back to the cannabinoids again. This paper is showing that the cannabinoids can help to protect you from misfolded proteins. But I see a link to deuterium-depleted water here because amyloid beta accumulates in the plaques. And when it's inside neurons, it increases the production of leukotrienes by this lipoxygenase. So this is going to be deuterium-depleted water because these lipoxygenases are fantastic at producing deuterium-depleted water. And when the leukotrienes are there, they increase A beta accumulation in a feedback loop. So I think the cell is aware there's too much deuterium. It's got different ways to remove it. One of them is to use lipoxygenase to pull it out of fats. Another one is to trap the deuterium in this plaque. It's using these methods to reduce its deuterium level. And the cannabinoids, I think, may catalyze the fibroproteins to make them better able to work properly to do the job that's needed to deliver the mitochondria, this healthy deuterium-depleted water. So the endogenous cannabinoids derived from arachidonic acid would do the same thing, but the CYP enzymes that are needed to synthesize them are blocked by glyphosate and other toxic environmental chemicals. I just want to show this picture. Glyphosate usage on corn and soy is highly correlated over time with deaths in dementia. This is one of Nancy Swanson's plots. The red line is the glyphosate usage. The yellow is the deaths per 100,000 of Alzheimer's and dementia. And so you can see that glyphosate is highly correlated with the rise, and there is an enormous rise in dementia over time in our country. Prion diseases and their biochemical mechanism. This is the CJD disease. That's a form of mad cow neurodegenerative disorder caused by prion protein misfolding. And ingested prions can see the disease process. So this is so fascinating because what can happen is that you've got a bug in your gut, some kind of pathogen is growing in your gut. It produces a prion-like protein. And then that prion-like protein is taken up by the immune cells in the gut, and it's brought into the gut-associated lymphoid tissue, and then it's carried into the spleen. So the spleen accumulates misfolded or potentially misfolded prion-like proteins from an infection, and the immune cells in the spleen are trying to deal with that. And if they can't clear it, and this could be because of their lack of energy, because they've got mitochondria that aren't working well, they will ship it out in the form of exosomes. And so what happens is the immune cells in the spleen and in the gut release these exosomes packaged up with this misfolded protein. And the protein travels along retrograde nerve fibers from the splanchnic nerve and from the vagus nerve, and they make their way up to the brainstem nuclei. So these exosomes are being ditched by these cells because these cells are unable to deal with this misfolded protein that they're picking up from the infection. And then the neurons in the brain take up the misfolded proteins, and then they can trigger the proteins that are already in the neurons to also misfold. This is the whole crystal-like effect of these prion-like proteins, that they can cause other proteins to join the club, which can really cause a cascade effect that is so damaging to the cell. And so it's a terrible situation. And this is just a picture from this paper, rather complicated picture. This part I don't want to pay too much attention to. This is showing the inside of the gut area where you've got these immune cells doing stuff. But what's interesting to me is this part over here, the spleen and the gut and these efferent fibers of the parasympathetic nervous system and of the sympathetic nervous system. These fibers are carrying these exosomes along and these misfolded proteins. And this is a build out of this place right here on the nerve fiber. We've got these two cells, and these blue guys are the good guys. Those are the prion protein that's not misfolded. The red ones are the misfolded ones. And what's happening is both here and here and here, these the misfolded protein is causing the good protein to also become misfolded, and then you get more misfolded, and eventually you've got a bunch of them showing up at the nerve, at the brainstem, all of these misfolded prion proteins, prion-like proteins delivered to the brain. And so the disease process is starting in the gut and the spleen. And then after many, many years or even decades, in fact, it finally gets to the brain, and the brain gets loaded with enough of these misfolded proteins that you start to show symptoms of neurodegenerative disease. So deuterium isotope effects on lymphoid tissues and humoral antibody responses in mice. This is I was really happy to see this paper, and it's hard to find papers on this sort of thing. They're very rare. This one is from 1979. So they're old also, many of them are old. And where they were doing these experiments of exposing animals to deuterium-enriched water. They're not doing these experiments anymore. But this one was quite interesting to me because it showed, first of all, it disrupted their immune system. It depressed their antibody response to toxins. And it also affected very strongly the thymus and the spleen and the lymphocytes. And then the white pulp of the spleen, the thymus, the lymph nodes, and the gut-associated lymphoid tissue. So all of this lymph system stuff was badly messed up by the deuterium, more so than other parts of the body. And the spleen was especially sensitive to the deuterium compared to all the other organs. So I think the spleen is a very interesting organ that I've been studying a lot lately. And I think it's very strongly connected to maintaining low deuterium in the blood. And the spleen, of course, is loaded with blood. It's got a really strong vascular system. It's a place where the blood is cleaned. It's a cleansing, clears the red blood cells. But I think it also plays that role of maintaining, supplying deuterium depleted water to the vascular system. So alpha-synuclein in Parkinson's disease, and this is sort of a great paper that also tells this same story. When the autophagic mechanisms fail to digest the toxic oligomers, and what this means is that the lysosomes are broken. The lysosomes are not working properly in these dysfunctional cells with this, I think, with excess deuterium. So these cells have to get rid of this misfolded protein or else they're going to die. And so they release them in the form of these exosomes. And that could be a way to clear the cells of the toxic species. This is a quote from this paper. Unfortunately, allowing uptake of these exosomes by neighboring cells. So the cells are ditching the toxicity that they're carrying, and then other cells are picking them up, and now they are presented with a problem. And so you find a cell that's healthy enough to clear it, that's going to be great. The problem is solved. But if too many of the cells are sick, then you just can't deal with all these misfolded proteins in the right way. Alpha-synuclein binds to membranes in its N-terminal domain. It has a C-terminal domain that is intrinsically disordered, and that's the kind of domain that you see in all of these misfolding proteins. They have what's called this intrinsically disordered domain, where the protein doesn't fold into either alpha helices or beta sheets. It just kind of hangs out in the cytoplasm, picking up deuterium. It has an opportunity to pick up deuterium. And so if there's too much phosphate, so there's a lot of phosphorylation cascades that happen in response to inflammation, they end up phosphorylating these proteins. And the other problem is if there's too much expressed, so different kinds of stresses can cause the cell to overexpress these proteins. They become too crowded, they become phosphorylated. That's what's going to cause them to form these soluble oligomers. And then those oligomers can block access of the hydrophobic domain to water. And this is very, very important, because when you get those oligomers, you've trapped the deuterium in the oligomer, and it can no longer exchange with the water. So this is an interesting paper giving a hint at alpha-synuclein's toxicity, that it could be due to its inability to function in its proper role. This is a loss of function toxicity for alpha-synuclein. What happens is alpha-synuclein plays an important role in managing dopamine. Dopamine is what's connected to, dopamine toxicity is what's connected to Parkinson's disease. And dopamine, of course, is a neurotransmitter that's produced in the substantia nigra as a signaling molecule for movement and things like that. And dopamine is normally sequestered inside these vesicles, and alpha-synuclein goes into the membrane. Here's an alpha helix alpha-synuclein molecule in the membrane that then promotes the uptake of the dopamine into those vesicles. And when alpha-synuclein misfolds, it can come off the membrane when it gets phosphorylated. And then it no longer can help the dopamine to get in. And so the dopamine stays outside in the cytoplasm. And now the dopamine is a very reactive molecule that can cause reactive oxygen species through these hydrogen peroxide. These kinds of reactions over here are releasing oxidizing agents that are going to damage the cell. So when the alpha-synuclein is not working properly to let the dopamine get into the vesicles, the dopamine becomes toxic. And then here's another picture from that same paper. Defective sequestration of dopamine into vesicles, leading to the generation of reactive oxygen species in the cytoplasm, is a key event in the demise of dopaminergic neurons in Parkinson's disease. Quite a nice quote there. And this is showing sort of the oxidative damage, because the dopamine is damaging the lipids and the DNA, and the cell is getting very sick. Whereas if it has enough defense mechanisms to protect from those oxidizing agents, it'll be okay. And these are all these reactions that are going on here with the dopamine. So the dopamine is actually a good source of deuterium depleted water. And so what's very interesting to me is that when the alpha-synuclein misfolds, the dopamine sticks around the cytoplasm. It starts doing all these reactions. It starts producing deuterium depleted water while the alpha-synuclein is busy trapping deuterium. So both of them are working towards reducing the deuterium load in the cell, trying to recover from the toxicity of the deuterium. This is what I'm thinking is going on. So here's my hypothesis. Prion-like proteins are normally intrinsically disordered. They're hanging out in the cytoplasm. They're picking up, they're exchanging freely with the deuterium that's there. If there's a lot of deuterium in the water, they will misfold. They will form into these beta sheets that are going to trap that deuterium and no longer allow it to freely exchange with the water. So it's removed deuterium from the water. They can bind to the membranes in order to gain access to structured water, the gelled water, which is where deuterium is enriched and the pH is elevated. Both of those are going to help to cause it to trigger into that misfolded form if there's too much deuterium there. They spontaneously organize into these oligomers under conditions of high pH deuterium-rich exposure of, I think, low pH. That's a mistake. In conditions of low pH. No, that's right. High pH deuterium-rich exposure, in structured water. So the excess bound deuterium hydrogen ratio facilitates this refolding. This is the point, that when there's too much deuterium in the water, the protein misfolds, it traps the deuterium in the fibrils, precipitates them out, and serves to lower the deuterium levels in the cell. And when there's an impaired ability to clear the misfolded proteins, they are shipped out as exosomes, and then they can be taken up by other cells, which might be better equipped to degrade the prions. And once there's not enough cells around that can degrade them, then you're going to get disease. So researchers have been, so I'm just returning this slide just to summarize all of that. Researchers have been unable to figure out exactly how misfolded prion-like proteins cause disease. I propose they're looking under the wrong light. Lewy bodies and plaque are an indicator of impaired ability to maintain low deuterium levels in the mitochondria and the lysosomes. The proteins misfold when deuterium levels rise too high, and their misfolding works to deplete the deuterium, helping to solve the problem. But then an inability to clear the misfolded proteins can be caused by insufficient heparan sulfate in the membrane glycoproteins, which in turn is caused by the glyphosate exposure. The plaque regions are the solution to, not the cause of, the toxicity. So now I want to get into G quadruplexes, which are really fun, and I've only started learning about them in the last couple of months. Something brand new to me, intricate coding that goes on with the prion proteins and the G4s. Interesting, really interesting coding. So guanosine is one of the nucleotides in the DNA. DNA has these four-letter code, and it is able to form gelled water. And it can do that when it forms these G quadruplexes. So this is so interesting because the guanines, four guanines get together to form a structure, and then those joined up with other guanines, you get a bunch of guanines forming a structure that creates structured water around it, gelled water, which is going to be trapping deuterium. And so this is the prion protein, which binds to these G4 RNAs, destabilizes its structure, and triggers the conversion of the protein to the misfolded form of protein. So this is a healthy prion protein. This is a misfolded prion protein. When the protein binds to a G4, the RNA, that's the RNA in a G4, it causes the G4 to fall apart, but it also causes the protein to misfold. Both of them happen simultaneously when the prion protein binds. The messenger RNA that codes for the prion protein, has several of these G4 motifs located in a particular region called the octarepeat region of the protein. So the protein binds to its own RNA at these G4s, and when that happens, both the protein and the RNA are affected. And so our results allowed to surmise a quadruplex unwinding activity of the prion protein that may have a feedback in vivo. So this is just really, really fascinating stuff to me about the prion proteins and how they interact with their own RNA. And in doing so, they alter both the RNA and the protein. And I think the whole game here is to sweep up deuterium and get rid of it. So here's the intricate coding of these G4s. It's really fun. You have this GXXXG motif, which is a signature motif of prion proteins. These two Gs are glycines, two glycines spaced by 3 wildcards in a regular order over a long sequence of a prion protein. So glycine DNA, the code for glycine in the DNA is GGX. This is guanine, and then anything. So GGG would code for glycine. 2 guanines followed by any nucleotide. Tryptophan, which is called W, its code is UGG. That's uniquely tryptophan. 2 Gs at the end, preceded by U. GGGWG, so this is 3 glycines, a tryptophan, and then a glycine. That's the motif that reappears multiple times in these prion proteins, GGGWG. Glycine, 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 tryptophan, glycine. That is going to be here in the DNA, GGX, 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 UGG, GGX. So you've got 4 glycines in a row here, and you've got all these glycines. All of these can buddy up to form this structure in the DNA. These are the guanines, these blue dots, and they have a metal in the middle, like potassium. And you have this nice structured organization that creates gelled water around it in these proteins, in the RNA that's corresponding to these proteins. So just to show this again, in this G quadruplexes within prion mRNA, is this the missing link in prion disease? This is an article that I found, which I was delighted to see, because I'm so fascinated by this ability of the protein to interact with its own RNA, I think, in order to trap deuterium and remove it from the cell. And so here's the sequence in the protein, in the prion protein. This is the GGGWG that's occurring multiple times in the protein. And then this is the DNA that corresponds to it. You can see these DNA sequences with all these Gs. And then these guanines, these are going to form these structures, and the structures are going to buddy up to form a G quadruplex. And then those are going to gel the water. And then the prion protein that's produced is going to stick to that, to the G quadruplex in the gelled water and pull out the deuterium from the gelled water. And so here you can just see that that's a GXXXG motif here going for a very long time in these prion proteins, characteristic feature of prion proteins. So there's a code both in the protein and in the RNA. I just want to remind you, there's a paper that I wrote together with colleagues, does glyphosate acting as a glycine analog contribute to ALS, Lou Gehrig's disease? And so we looked at certain proteins, this TDP43 and also this FUS, fused in sarcoma. Both of these proteins are very interesting. They're RNA binding proteins. And TDP43 has this long glycine-rich region right here. You see all these mutations. You can see all these mutations here. Those are, many of them are glycines getting converted to something else. When you have those mutations, you have an increased risk to ALS. Glyphosate is going to have a field day with this sequence because there's so many glycines in here that it can substitute for. TDP43 has a long glycine-rich region, many mutations linked to ALS. It binds to G4s in multiple RNAs, and fused in sarcoma also binds into this RGG domain. So all of these proteins that are linked to ALS have the same property as the prion proteins of binding to RNA, and I suspect removing deuterium from the cell's cytoplasm. Whenever a protein binds RNA, it's binding to phosphate, and phosphate binding sites are typically enriched in glycine. So this is going to give you the glyphosate susceptibility motif, and glyphosate is going to be able to do a lot of damage to these proteins. Now, this is just fragile mental retardation protein, a protein that is misfolded. Mutations are associated with mental retardation. And I just want to show this RGG segment of it, and you can see all these Gs. These blues are all Gs. So this has tremendous opportunity for glyphosate to cause trouble. This paper is about this particular glycine 11 right here. It shows it over here in the structure. This is binding to a G4. And when you have a mutation, you greatly reduce its ability to bind. If glycine is not glycine at 11, tenfold reduction in the binding affinity, which makes sense because it's blocking the ability to fit into the phosphate. Glycine to glutamate mutation in this protein causes Fragile X syndrome. Now, I liked this paper because it has a quote here. It showed that there was a mutation at 266, glycine to glutamate, and it was unable to bind the RNA, which you would expect. And this is what it said in the quote: structurally position 266 likely requires a small, flexible, and nonpolar amino acid, all the characteristics of glycine. Glutamic acid, which is glutamate, on the other hand, large and negatively charged, and is predicted to clash sterically and ionically with the surrounding amino acids. And so that's perfectly a description of glyphosate as well. Glutamate is similar to glyphosate, and I looked for glutamate mutations to try to find potential proteins that could be affected by glyphosate, because glyphosate is also a bulky, negatively charged amino acid, just like glutamate. So I think this makes a lot of sense that glyphosate would also cause something like Fragile X syndrome without a mutation, just because glyphosate is actually substituting for glycine during protein synthesis. Whoops, I missed it. About a third of individuals with Fragile X syndrome have features of autism, such as problems with social interactions and delayed speech. So the big picture, reviewing this, do prion-like proteins play a role in maintaining low deuterium in the cytoplasm? And my hypothesis is that they can trap deuterium inside water and accessible precipitates. Bold claims, misfolded proteins in Alzheimer's plaque or Lewy bodies are a sign of impaired deuterium homeostasis. Prion diseases begin with stressed immune cells in the gut and spleen. Cells with impaired ability to clear misfolded proteins release them inside exosomes and deliver them to other cells, propagating the disease. Impaired sulfate homeostasis plays a significant role. So now I guess I'm running short on time, so I better get hustling. Just realized people who suffer from acute syndromes from COVID-19 are immune compromised due to system-wide defective mitochondria, I believe. The virus invades the lungs and proliferates there, inducing an inflammatory response. This allows lipids in the viral membrane to be metabolized by lipoxygenase, which produces deuterium-depleted water. Sharp upregulation of bradykinin results in a dramatic drop in blood pressure, infiltration of water and macrophages into the lung alveoli, massive overproduction of hyaluronic acid. A hydrogel encasing the hyaluronic acid fills the alveoli and traps deuterium, and the signaling cascade causes myeloid cells from the bone marrow and the platelets to come into the lungs and release their mitochondria into the medium, which is rich in deuterium-depleted water. They supply the mitochondria, handed off to the immune cells, supplying them with new mitochondria that are healthy, along with deuterium-depleted water. So the whole process that's going on in the lungs in response to COVID-19, I believe, is a mechanism to supply deuterium-depleted water and healthy mitochondria to the immune cells in order for them to be able to fight off the virus. This is a paper that I was really happy to see, pathogenesis of COVID-19 described through the lens of an undersulfated and degraded epithelial and endothelial glycocalyx. So this is the sulfation defects in the heparin sulfate proteoglycans, which could play a major role in the pathogenesis of COVID-19. The intact, highly sulfated glycocalyx inhibits viral infection. Undersulfated glycocalyx leads to dysregulated immune response and subsequent cytokine storm, and many toxic environmental chemicals disrupt the sulfate supply. And this is a diagram of the various movements of sulfur through the sulfur system. These are sulfur-containing amino acids like cysteine and sulfite. But across the top here are two critical enzymes, the PAPS synthetase and the, it's hiding there, sulfotransferase. PAPS synthetase makes PAPS, sulfotransferase produces the sulfated product. So you need both of those enzymes, PAPS synthetase and sulfotransferase, to make to add sulfate to the glycocalyx. And that's what's defective in autism, which causes heparin sulfate deficiency in the brain, which disrupts neuron development. Both of these enzymes bind phosphate at sites that have highly conserved glycine residues in this GXX-GXGK motif. So this is critical because I think these are being disrupted by glyphosate. And I think glyphosate is a major cause of the bad outcomes we're seeing in the United States with COVID-19. Sulfate maintains the gelled water lining the walls of blood vessels. It traps deuterium, leaving the fluid blood plasma depleted in deuterium. So this is just a diagram of the immune system. You have the innate immune system and the adaptive immune system. The innate immune system is unhealthy in the presence of glyphosate because its mitochondria are not functioning well. It can't clear the virus, and then it needs to be repaired through a process involving depletion of deuterium in order to restore the health of the macrophages so that they can clear the virus. This is a mouse study that showed aged mice have dysfunctional T cells, and they have defective mitochondrial oxidative phosphorylation, causing an inflammatory response. They had a weak immune system. They died from an infection with the pox virus much more readily than the young mice. And they call this inflammaging as a process of growing defects in the innate immune system as you age, with all these different diseases that are associated with it. Initial COVID-19 events, SARS-CoV-2 viruses enter through the bronchioles and infect epithelial cells lining the alveoli. The macrophages fail to clear the virus because they're defective. The virus proliferates wildly. The macrophages send out alarm signals, which brings in these additional immune cells. The capillary walls become leaky to support the invasion. So the macrophages all come into the lungs here. Blood pressure drops, fluid begins to accumulate in these spaces. And that's what gives you this inability to breathe. You can't get enough oxygen. The person feels as if they're drowning. So this paper is a bradykinin storm brewing in COVID-19. And it points out that hypertension is a risk factor, but hypotension develops during the disease process. And you have this interaction with the ACE2 and the ACE receptors, where the ACE2 receptor is actually greatly upregulated, and ACE is downregulated. ACE is what normally degrades bradykinin. So and also bradykinin receptors are upregulated by a huge amount. Bradykinin induces vasodilation and hypotension, which is what draws in all this fluid. And you introduce these inflammatory cytokines, which cause the capillaries to leak and inhibit fluid reabsorption, leading to this alveolar flooding. And here's a picture here of this alveolus filling up with this high hyaluronic acid, which makes the hydrogel, which traps the water and traps the deuterium as well. So this is a way to trap deuterium in this hydrogel, leaving behind deuterium depleted water that these macrophages can take advantage of. There's a massive overproduction of hyaluronic acid in the lungs and associated with severe disease. It can trap roughly 1,000 times its weight in water. And then when it's bound to the water, it causes a hydrogel with a stiff, viscous quality similar to jello, filled with deuterium. Multiple enzymes that synthesize it are massively upregulated in the COVID-19 lungs, incredible amounts, 9,000-fold on HAS1. So you're making lots and lots of hyaluronic acid, you're making lots and lots of gelled water that's trapping deuterium. You have these ground glass opacities, and the hyaluronic acid in the bronchial space of the lungs could form a viscous hydrogel that would negatively impact gas exchange. So you're not able to get oxygen, but you are making a tremendous effort to get rid of the deuterium in the water. So this is how I see this story. The virus contains, well, here's another piece of the story, is the virus. It contains these lipids, linoleic acid, that comes in from it steals it from the host cell. Creates this inflammatory response. It starts releasing lipoxygenase, which can produce deuterium depleted water. The leukotrienes are powerful signaling molecules. We saw this before. The arterioles constrict, the venules open up, the water pours into the lungs, and it is a healthy water with low deuterium that can resupply the mitochondria with much needed deuterium depleted water. And this empowers them to clear the virus. And as you remember, lipoxygenase has a fantastic ability to select hydrogen over deuterium in making water. It makes water. So the virus, here's just a picture of the virus trapping this linoleic acid. It fits perfectly into the virus membrane. It picks it up from the host, and then it gets it modified through the lipoxygenase to produce the deuterium depleted water. The patients that have severe disease have been shown to lose fatty acids. They have low levels of fatty acids because they're being consumed by these lipoxygenase. Viruses are stabilized by excess deuterium. And this is a paper that just shows that of all viruses, not just these COVID viruses, they take up deuterium, they trap it in their protein code, and they trap it in their single strand of RNA. And the deuterium actually stabilizes the virus and protects it from degradation from high temperatures. So the body raises a fever, produces a fever to try to break the virus. But if there's not a deuterium, the virus is sturdy and it can survive better. So that's a very natural way in which the virus will be able to proliferate under conditions where deuterium is in trouble. And then the body can invoke this entire process to try to fix the mitochondria in the immune cells in order to be able to fight off the virus. And this shows about the exosomal transfer, which is quite fascinating. You can actually use these exosomes to transfer mitochondria from one cell to another. And you have these cells that are coming in from the bone marrow, precursor cells from the bone marrow that can come into these inflammatory sites. And then they drop off their mitochondria, they release them in these exosomes, and then the immune cells, like the T cells, the macrophages, the dendritic cells, can all pick them up. So you've got these wonderful mitochondria from the bone marrow being supplied by these bone marrow cells to the immune cells, along with the deuterium depleted water. So they're really fixing themselves up with all of this water and mitochondria through this intercellular communication. Coronavirus, sepsis, revisiting mitochondrial dysfunction and pathogenesis, aging, inflammation, and mortality. This is a paper that's talking about depleted glutathione, damaged mitochondria, which upregulates this HIF1 alpha, and depleted NAD plus supplies impede the recovery phase. This is, again, this really important nutrient that's supplying the deuterium depleted H. And you get sepsis, and then you get a hypoxia, low oxygen, blood clots, lactic acidosis, multiple organ failure, a nasty consequence of deficiencies in NAD plus, which of course are connected to problems with deuterium. Hemoxygenase and COVID-19. So some cases cascade into a hyperinflammatory state that leads to thrombosis and multiple organ failure with this upregulation of HIF1 alpha, which upregulates hemoxygenase, which normally would tame the inflammation. And the interesting thing about hemoxygenase is that it modifies heme. It produces biliverdin from heme, which is coming from the red blood cells. And every time it makes biliverdin, it produces 3 molecules of deuterium depleted water from oxygen and NADPH. Very, very important. So I think this hemoxygenase is a mechanism, again, to produce deuterium depleted water. However, glyphosate can get in the way here as well, because it's a critical glycine residue in hemoxygenase, which if you change it to something else, it breaks the molecule such that it can no longer convert heme to bilirubin. So it doesn't produce a GDW, and it also releases ferrol iron, which is a very toxic form of iron that reacts with the reactive oxygen species to damage the vascular wall. So you get a positive feedback loop that's highly destructive. And this has been shown to be true. Enzymes, that mutations in this protein that don't have that glycine have this property that they're destructive, they're counterproductive, and they increase the oxidative stress. So to recapitulate, innate immune system in the lungs is impaired due to chronic exposure to glyphosate and other toxic chemicals. Macrophages can't trap and clear the viruses due to defective mitochondria. The virus proliferates and causes a strong reaction from the adaptive immune system. The inflammatory response leads to a complex cascade of a cytokine storm, eventually damaging the circulatory system. Heme oxygenase is upregulated. It would normally tame the inflammation, but glyphosate turns it into a rogue version of itself. This planned response has a goal of temporarily shutting down the mitochondria so that they can be resupplied with deuterium-depleted water obtained through direct assistance from the viruses, and it backfires when many of these proteins are disrupted by glyphosate. So finally, healthy lifestyle. A high-fat diet is a low deuterium diet. Butter, lard, and coconut oil were among the foods with the lowest levels of deuterium among foods tested. The synthesis of fats, as we've seen, involves the transfer of hydrogen from NADPH into the growing fatty acid chain. So just showing you the amounts of deuterium in various foods. Flour and sugar have high levels. It comes down for cottage cheese, lower for olive oil. Butter, ghee, and coconut oil are very low, and lard, pork lard as well. These all have much lower levels of deuterium. So a high-fat diet is a low deuterium diet. This links deuterium to mast cell activation syndrome, and the mast cells release histamines during an allergic reaction. Deuterium increases the histamine release from the mast cells. And plants get rid of deuterium by storing it in sugar and starch. So fruits and grains are high in deuterium. Meats from grain-fed animals are also high in deuterium. Leafy green vegetables, animal fats, lard, tallow, and butter, and plant fats, avocado, coconut, and olive oil are all low in deuterium. And grass-fed beef and the dairy products derived from pastured cows are excellent sources of low deuterium fat. Oops. Interestingly enough, waterfalls are therapeutic, and that's quite, they've shown that in this study. They actually showed that they were healthy, hanging out near waterfalls. And these actually produce negatively charged nanowater particles known as Leonard ions. And what I think is happening is these particles are deuterium depleted. Just as I mentioned, when the rain comes down, the water evaporates, it's low in deuterium. The water that's released from these in the mist is also low in deuterium. So when you breathe that water, you're getting low deuterium water through the lungs. And so they talk about added value of exposure to waterfall microclimate. Ocean waves probably have a similar effect. I think it's interesting to look at Iceland. Life expectancy in Iceland compared to Europe and to the United States. In Iceland in 2015, its infant mortality was 1.9 per 1,000 births. The European Union was 4.0, and the US had an infant mortality of 5.87. Really shocking difference, 1.9, 5.87. They have a lot of good stuff in Iceland. They have these glacier water, and they have ocean waves to give you, hanging out by the ocean near the waves, you're getting that same effect as you would get from the waterfalls. And they have these birds that they eat a lot of smoked puffin and various kinds of seabirds that are very healthy fats, healthy deuterium depleted fats. So they've got lots of opportunity. They also have a lot of sulfur. So this is black sand beach. This black sand is loaded with sulfur. So their soil is sulfur rich, and that's helping them to maintain health. This is a book called The Jungle Effect. It looked at different places in the world where people had really high life expectancy. Iceland is certainly not a jungle, but it was one of the places she studied. Their diet consists of fish, sheep, seabirds, potatoes, and other simple vegetables. It has glacier water, which is low in deuterium, and it has basalt, which is high in sulfur. And I think both of those are crucial for maintaining the healthy diet. Sulfur hot springs have many therapeutic benefits. And it's interesting, after an eruption in the 1800s, many of the islanders moved to northern Canada, and they lost their health benefits despite continuing to eat a similar diet. This is just a diagram of all the different ways in which B vitamins are important for all this maintenance of this H on the NADH in the mitochondria. This is the complex one. These are the complexes of the mitochondria, and just lots and lots of B vitamins, zinc, and vitamin C. These are all really important to maintain healthy mitochondria. So it's really important to eat foods that are naturally have dense in vitamins and minerals. eat foods containing sulfur. These are some good sulfur containing foods: fish and eggs and cheese and seafood, onions and garlic, and cruciferous vegetables. Supplemental sources of sulfur. These are some of the things you can take as supplements, I think, that contain sulfur. And it's interesting, you can get these glucosamine and chondroitin sulfate. Glutathione has 3 amino acids, one of which is cysteine, which is sulfur containing. You can take cysteine as a supplement. Alpha lipoic acid has sulfur. Taurine is an interesting amino acid that contains sulfite, dimethyl sulfoxide, and methyl sulfonylmethane, methionine, acetylbenzoyl methionine, and then Epsom salts. I like Epsom salt baths as a way to get sulfate through the skin. They can have many beneficial effects. They're nearly non-toxic, and Epsom salt baths is my favorite. Foods high in niacin. So niacin is the precursor to NAD, and these are some of the good foods that you can eat to support niacin: avocado, peanuts, whole grains, mushrooms, green peas, and potatoes, and also meat, fish, and poultry. Riboflavin is a source of FAD. So riboflavin is a B vitamin, B2. It supplies FAD. And again, these are the foods that are high in riboflavin, all very healthy foods: mushrooms and green vegetables and nuts and avocado, fish and eggs and milk. You can drink deuterium depleted water. I have some here. Light water is what I drink. You can buy a big jug like this, and you only have to take a little because this is very low in deuterium, only 10 parts per million. And you mix it with regular water, like 2/3 of regular water, 1/3 of this. So you just use a little bit every day, like 1/3 of a cup, to provide yourself with something that's similar to glacier water. Hydrogen water, I suspect, is also a source of low deuterium. Hydrogen, again, the same principle that the hydrogen atoms that are made out of the water, the hydrogen atoms, H2 molecule, is probably going to be low in deuterium, and you get that through the hydrogen water. Flavonoids and polyphenols, these are basically brightly colored fruits and vegetables are good sources of flavonoid, and also herbs and spices, all good sources of flavonoids and polyphenols. And then, of course, go organic. I can't emphasize that enough. This is a picture of my kitchen table. When we shop at the grocery store, we always buy organic, certified organic food, or if we have a trusted farmer, we will buy from the trusted farmer, small farm. I think it's one of the most important things you can do in order to reduce your glyphosate load and to keep yourself healthy. So organic, whole foods, rich with micronutrients, that's the path towards good health. So in summary, deuterium is pervasive in the environment. It's very damaging to the ATPase motors in the mitochondria. Biological organisms have devised sophisticated mechanisms to deal with it. A class of enzymes called flavoproteins have amazing skills to select hydrogen over deuterium in their product, but the enzymes are unfortunately disrupted by glyphosate. Heparan sulfate proteoglycans play an essential role in supplying protons to mitochondria and by trapping deuterium in the extracellular matrix. Prion misfolding diseases are likely caused by an inability to maintain low deuterium in the organelles. SARS-CoV-2 induces a reaction in the lungs that supports renewal of mitochondrial health to the immune cells. Glacier water, foods rich in niacin, riboflavin, healthy fats, and organic diet will all help protect you from deuterium toxicity. And thank you very much. I actually managed to get done in almost the right amount of time, so we're good.
Yes, indeed. A great job. Thank you so much, Stephanie. Just so much information, so thorough. We expect nothing less, but thank you so much for delivering all of that today. And so, yeah, if you've got time, we'd love to get to some live questions.
Great.
Excellent. Thank you. Actually, before we do, remind us all, where's the best place to get your books?
Oh, right. Yes. Well, Amazon, I suppose, is the best place. But Toxic Legacy is sold on several different bookseller sites and also from the publisher. But Amazon is probably the best bet.
Excellent. Thank you so much for that. Really appreciate it. So with that said, yeah, let's jump in. I see the hands being raised already. And as you know, that's how we do it around here. We generally don't take questions directly from the chat box. We try to have people raise their virtual hands. For our audience members that aren't sure how to do that, you have different functions in your Zoom window. One of those tabs is called your Reactions tab. You click on Reactions, you'll see different emojis pop up. One of them says raise hand. you'll click your raise hand button and we will see your hands raised. So after that, I'll call you by your first name and I'll unmute you. And so Stephanie, if you're ready, I've got Sophia ready to go.
I'm ready.
All right. So Sophia, I'm going to unmute you now, and welcome, Sophia.
Hi, thank you so much for this brilliant lecture. It was really amazing. Please, my question is, what would help mitigate the side effects of a land where nearby lands glyphosate was spread? Is it better to grow veggies inside elevated, like wooden boxes, to attenuate the damage caused by glyphosate? Also, is using new organic earth is an efficient thing to do?
New organic what?
Earth, like where to grow veggies and so on.
Yeah, I think you're saying if you're living near a field where glyphosate's being used, is that what you're saying?
Absolutely, yeah. Please.
Oh, that's terrible. I don't know what can be done other than to move. I really don't. It's, it gets released into the air. It's I think it's very bad to live next to a field where glyphosate is being used. I would say move. I mean, I don't know how you could grow your own organic vegetables in that circumstance, unfortunately.
And was there a second question there, or was that everything, Sophia?
Yeah, it was okay. So growing veggies wouldn't be a good idea on this land.
Yeah, I don't think so. I mean, I think it's going to be very hard for you to keep your vegetables healthy if they're sitting there where the glyphosate's being sprayed next door. If it's very close, I think it's dangerous.
Thanks very much, Stephanie. Let's go now to David M. Welcome, David.
Thank you. I live in Canada, and the province of Ontario generates a lot of electricity using nuclear power. And the Canadian reactor design uses heavy water or deuterated water. And I'm wondering,
Interesting.
A lot of what you said went over my head, but I think your thesis is that deuterium is bad for you. Now, there would have been workers working in a facility to enrich the water, and the deuterated water is actually 99.9+% heavy water with the deuterium ion. Yes. I'm wondering if you have or will be looking at the health of these workers over the last 20 or 30 years to try to validate what you're saying.
That is a very good point, and that is excellent. I hadn't really thought about that, but that makes a lot of sense. And I'm aware that that's how they learned about deuterium, heavy deuterium water. They learned how to make it back in World War II, with the atom bomb and whatnot. So nuclear energy depends upon heavy deuterium, heavy water. And if I don't know what happens to that heavy water in the plant once is it probably just released into the circulation, and whether that impacts the amount of deuterium in the water that people are drinking. I don't know in the area. That would definitely be something that should be studied. I think American researchers are not really aware of deuterium. There's very little studying going on with respect to deuterium in this country. Most of the work is going on in Russia, Ukraine, and Hungary. Those are the three countries where all of this research emerged from those countries. And it's interesting because Russia, they learned about it because they had these people living up north in Siberia who were incredibly healthy. They were living to be 120 years old. I mean, people had very good longevity. And their diet was pretty plain because they lived up north. They didn't have a long growing season. And they couldn't figure out why they were so healthy. And they finally traced it to the water. And they finally figured that out, that they had this low deuterium water from the glaciers that was keeping them healthy. So that was really quite amazing to me. But yes, I think it's an excellent point, and I don't know, and I haven't looked, so I really should see. Do you know whether there's health issues with the people who work at the plant?
Well, I know that there was a facility in operation in Ontario for many years, 25, 30 years, and it's fractional distillation, and they would draw off the heavy water. I know that in each facility there was a health physicist present. Mm-hmm. Now, as far as I know, there was no thought that the deuterium itself was harmful. So, mm-hmm. But there would be health records mm-hmm. for all of those people, kept in the provincial health registry system. So I think you could probably find out, doing a little digging, if these areas of the province, and it's interesting where the heavy water enrichment facility was a farming area. So you'd probably have glyphosate as well.
Wow,
so you
that's
might
really
have that,
dangerous.
that you might have that effect synergy. But I think, health records would be available, and that would be a way to dig deeper into the health effects if you could find some patterns there.
Yes, interesting. Thank you.
Oh, okay.
Thanks very much, David. And David, I somehow I lost you, so I just want to make sure if you could hit your mute button too when you're done. Thank you so much for your questions. And now let's move on to Annette. Welcome, Annette.
Hello, I am not Annette, but I am using another person's Zoom because my computer didn't work. I am Ivan from Mexico, and I have several questions, Dr. Seneff. First of all, I want to thank you because you answer all my emails with very good information, and not much people do that. My question is about proteasomes that disassembles proteins in the body, and they are, I heard a study about the Viagra that activates proteasomes at the level that the people that use Viagra as their continuous medicine, they develop 69% minus Alzheimer than the rest of the population. And the other question is about protein rebuild, because when the protein is disassembled into amino acids, it builds again proteins in the body. And there is a cycle over there, but I cannot not understand the cycle until the the glyphosate is excreted. I personally, I have seen that the glyphosate is excreted through the skin at the last way to take it away from the body. But do you know something about that? And another question is about the use of chlorine to break down the molecule of glyphosate. Does it has toxic residues? Because I have heard about the toxic residue that AMPA, it's its name. And the other conclusion that I have made through your talk is that the people that has deuterium instead of water, they develop some kind of AIDS in their immune system. That's why this virus is so bad to the people, that they have some kind of AIDS, and that is not declared by their doctors until they became infected. That's all, Dr. Seneff, and I want to thank you again for your great work that really changed not only my life, but also the life of all the people that can read your book. Your book, it's so interesting. And I just wanted to hear you reading the book, because you share your emotions through your voice much better than the woman that read your book in the audio version.
I'm sorry that I didn't read my book. I should have.
Yes.
Thank
That's
you.
all, Dr. Seneff. Thank you very much in advance.
Right. Those are a lot of questions that you asked. I do think it's interesting with the glyphosate. I think it is possible that you could sweat it out through your skin, that you would push the glyphosate out into your skin, which could cause, skin issues, but would help you to get rid of the glyphosate. That's one way that you could get rid of it. I think you mentioned the chlorine dioxide. I think you were thinking of chloride and chlorine dioxide in particular. I'd like to bring that up because that's a very controversial medicine. It's a very simple molecule, chlorine and 2 oxygens, and it's highly reactive. It's oxidizing. It's like, you have ozone, for example. You have ozone treatment and chlorine dioxide treatment that some of the naturopaths promote as being health healing, and I believe that that's true. And it's interesting in particular because it's the opposite of what people keep talking about. You're taking antioxidants. People say you need to take lots of antioxidants, and that's like all these fruits and vegetables, the polyphenols and the flavonoids. and vitamin C and all these things that have antioxidant capability to protect from reactive oxygen species, whereas chlorine dioxide and ozone are actually the opposite. They're pro-oxidant. And so it seems very, very strange to take something that's pro-oxidant. And what people don't realize is that, redox reactions, oxidation reduction reactions are basic to biology. That's how everything happens. And in the mitochondria, you depend upon the oxygen to cause the reaction to happen, to be able to break down the food and make the energy. And so you have to have reactive oxygen in order for the body to work. But the problem with it is that the glyphosate is messing up those enzymes that would normally allow you to handle it perfectly fine. But because the glyphosate is messing up, for example, the succinate dehydrogenase, you end up with these reactive oxygen species in your mitochondria that wreck them. And so the trick, of course, is to avoid the toxic exposures so that your own enzymes work properly. But the chlorine dioxide is very interesting to me, and I suspect there's something to it. There's people who swear by it, who say it's fantastically helpful and also helpful for COVID-19. I've seen there's a nice documentary movie, a 1-hour movie, and I think the last third of it involves people who use chlorine dioxide to treat COVID-19 to good effect. And I think it's very interesting because, first of all, it's able to break down glyphosate non-enzymatically. So I think when you take chlorine dioxide, you're helping to remove the glyphosate in your gut. And but also it produces hypochlorite, and hypochlorite is an interesting oxidizing agent that can combine. Hypochlorite combines with taurine to produce something called taurine chloramine. And taurine chloramine is a reactive version of taurine, which can turn into sulfate. So I actually think that chlorine dioxide is a way to help your body make sulfate. And as I've shown, sulfate deficiency systemically is a major driver behind modern diseases. So I think chlorine dioxide could be a really good medicine to use under almost, for many different functions. And it's extremely controversial. The mainstream is saying that it's horrible because it's bleach. They associate it with bleach. It does have some relationship to bleach. And in fact, bleach is used in the water purification systems. They use chlorine. They use chlorine itself in the water purification system to kill the microbes, to kill the pathogens, to make the water safe. And the chlorine is actually very good also at breaking down glyphosate, even better than chlorine dioxide. It's not safe to drink chlorine. I would never recommend that you would take chlorine. But chlorine dioxide is a much milder version that is also produced, it's produced by the body itself as hypochlorite. So it's not, it's natural. It's a natural molecule. So it's not like a synthetic chemical like what the pharmaceutical drugs are.
Thank you very much for that, Stephanie. And thanks, Ivan, for all of those great questions. And let's go now to Mike. Welcome, Mike.
Hello, Stephanie. You lost me pretty much with, weed killer in your conversation. It was so intellectual. One thing I wanted to comment was that it just proves to me that during this conference, we talk a lot of the presenters talk about mechanisms. And I think you've proved to all of us that there's so many mechanisms that we don't understand or how they interact and so forth. And it's very important. And the question I have is, since you know all this stuff, do all of the people that are making decisions for food and farming and all that, do they know these same facts and they're just not wanting to believe them?
Good question. Yeah, I wish I understood, whether the folks at Monsanto and at Bayer, those people who are trying so hard to keep their chemical going, whether they actually believe it's safe or whether they just want us to believe it's safe. I wish I knew the answer to that, because it's possible they've been deceived. They're just not wanting to believe that it's not safe and therefore refusing to acknowledge it. I don't know. I'd love to know that. But to me, it's very obvious. And I think that the research is coming out more and more recently. Papers are coming out left and right in the last few years, ever since Seralini. I think Seralini was a watershed moment when he wrote the paper in 2012 that got retracted and then got republished, the famous pictures of the mice with the massive mammary tumors. I don't know if you've seen that, but kind of went global. And that was the first time that someone showed that glyphosate at very low levels, chronic exposure throughout the lifespan of a rat, would cause a lot of problems. And there was the mammary tumors, the kidney disease, liver disease, reproductive issues, all of these things showed up over time. But after the first 3 months, things didn't look so bad. I mean, couldn't really tell that there was a problem. So it's a slow kill. That's one thing I think that's caused them to be able to keep it on the market so long, because you have to do your study over a long period of time in order to recognize that there's a problem. And that's because it gets into the proteins and slowly messes them up worse and worse over time. And so you can get by with a short-term experiment that doesn't reveal the toxicity.
In fact, Andre Lou was with us a couple of years ago, last time we were in person. And if anybody wants to look on the Real Truth About Health YouTube channel, we have archives of that video where he goes into exactly what you just said, Stephanie, but on a much deeper level. If anybody wants to go look at that, just happen to remember that off the top of my head. Mike, thanks for the question. Let's go now to Ash. Hi, Ash.
Hi, thanks, Stephanie. Great, presentation. Can't wait to read your book. I was going to ask about chlorine dioxide as well. So that's great what you've said already. Just to be clear, though, if chlorine dioxide breaks down glyphosate, do we still need to do something to then get it out of the body? Like any binders, or there seems to be a big connection between using it with DMSO. I'm assuming that's because of the sulfate to help, the liver and whatnot clear it out. And if you know of any connection, I'm down a rabbit hole with thiamine deficiency. This seems to be an equally prevalent but hidden epidemic, and connected with sulfate deficiency and oxalate toxicity. So yeah, I wonder if you could speak to that. Thanks.
Well, that's interesting that you bring up thiamine, because that's one thing I've been studying recently. I've gotten interested in thiamine and thiamine deficiency in particular, and I think it is related to the sulfur problem, because I talk about hydrogen sulfide gas. Like the autistic kids have a lot of bloating in their gut, and they have excess hydrogen sulfide gas. They have an overabundance of desulfovibrio, which is a microbe that converts sulfite into hydrogen sulfide gas. And hydrogen sulfide gas actually breaks down thiamine. And so you can end up with a thiamine deficiency because the hydrogen sulfide gas in your gut is breaking it down. And the reason why you have the hydrogen sulfide gas is because you can't take the sulfate up to where it needs to go. And I have that one slide on the COVID-19 where I showed that pathway that involves the PAPS synthase, which involves combining ATP with sulfate to make a phosphoadenylyl phosphosulfate. It's called PAPS. That is the universal sulfate donor. And the enzyme that makes that, I think, is being suppressed by glyphosate. And then also the enzyme that transfers from that PAPS to another molecule, receptor molecule, to make heparin sulfate, to make cholesterol sulfate, to make vitamin D sulfate, to sulfate tryptophan, all these different biologically active molecules, all of the hormones, these things all get sulfated in transit. And the enzyme that puts the sulfate on gets disrupted by glyphosate. I think that is a major player in multiple diseases, and especially in autism. The autistic kids have been shown to have, a recent paper showed they had severe suppression of the activity of this sulfotransferase, which is what attaches sulfate to things. And that's also going to disrupt the ability to clear metabolites that are produced by the microbes that are toxic. And then the body puts a sulfate on there to make them water soluble so they can get released through the kidneys, and that's getting disrupted. So you have an inability to make sulfate, which drives sulfite down towards hydrogen sulfide gas, which then can mess up the thiamine, break down thiamine and make it deficient.
Thanks very much, Stephanie. Now we have Joanna coming in. Hi, Joanna.
Hi, Stephanie. Thank you so much. I really appreciate your work, and I just really want to say keep at it. I think that interdisciplinary approach that you have to things is really adding quite a lot to this field. So thank you.
Thank you.
Just a quick note to Mike. I just wanted to flag that Baum Hedlund, a law firm that was involved in California litigation, released a lot of documents showing Monsanto does know and has known about the toxicity of glyphosate. It's funny because I thought I was going to ask you a question to which there was no known answer, and you started answering it in the last question.
Oh,
and my husband, who's had, health compromised for various reasons, over time he's developed intolerance to cruciferous vegetables, garlic, things like that.
There you go.
A lot of gas and things like that. And so you were talking about both kind of a that sulfite gas, which seems to be part of the answer, and then the thiamine deficiency. Is there anything else that you might recommend for somebody in the situation?
Yeah, I think glyphosate is actually causing an epidemic in sulfur sensitivity. And I discovered it actually when I was telling people you got to eat a lot of sulfur sources of sulfur, like cruciferous vegetables and onions and garlics. And people would send me email and say, no, I can't. I have sulfite sensitivity problem. And I discovered it from people who sent me email to share their problems with me. And I started looking into it. And that was early on when I was actually concerned about sulfate and autism before I even knew about glyphosate. And it's interesting because once I discovered glyphosate and then I realized that it would disrupt those sulfur maintaining enzymes, then I realized that would be what would be causing the problems that the autistic kids have. And many, many other people. Colitis is also connected to deficiencies in the sulfur system. It's so, so important. The sulfate is so amazing with all the things that it does for the body. And when you can't make that work, you just have an enormous number of problems every which way you look. And so I forgot where I was going with that, but yes, you're right. I think that we have an epidemic in sulfur sensitivity. And I suspect that if people were to be more careful to eat certified organic, they might actually lose their problem. The problem might go away because I think it is connected to the glyphosate.
He does eat non-organic wheat bread. So now I have something to tell him.
Yeah, the bread, I mean, that's the celiac disease. And I'm absolutely convinced that glyphosate is the primary cause there. Glyphosate in the wheat. And wheat is one of the foods that's sprayed with glyphosate right before harvest. And the celiac disease, we actually wrote a paper on glyphosate and celiac disease showing that the level of glyphosate in the wheat over time matches very well with the rise in celiac disease.
Mm.
What a special forum when we have a speaker that was featured yesterday is coming on and asking questions of another speaker today. Joanna, thanks for joining us today, Joanna.
Thank you so much, Stephanie. Appreciate it.
Thank you.
And thank you, Stephanie. And so let's now go to Alejandro. Hi, Alejandro.
Hi, Stephanie. Thank you for being here with us and for all the work you're doing for humanity. Really appreciate it. So just a couple of things that I wanted to bring to your attention that you might find interesting. So I've been listening to podcasts by a scientist named Mike Adams. You might know him.
I do know him, yes. Uh-huh.
And so he's been trying to bring a lot of awareness around some of the information that you've been talking about, especially for glyphosate, and the lab that he runs tests food for glyphosate. But anyways,
Right.
So he brought up a segment about a researcher named David Ostroff, who's an immunologist from University of Florida. He said he discovered two compounds that keep COVID-19 from replicating.
Oh, uh-huh.
It was diphenhydramine, which is the chemical in Benadryl,
Uh-huh.
which is an antihistamine, and then lactoferrin from breast milk found in colostrum.
Wow. That's pretty exotic.
Yeah, and then the other thing too that I wanted to mention is spike things that help keep the spike protein from attaching to the receptors, which is black seed oil and honey, pine tea, and star anise tea.
Yes, I've heard about that idea. Yes.
And then like those have to do with shikimic acid, right?
Oh, interesting. So
thank you. I was just wondering if you could kind of just maybe talk about those types of things and maybe, other helpful information around those subjects.
Yeah. I mean, I'm not a practitioner. I certainly do have practitioner friends who have been trying all of these things. And I think it is true that a lot of these, they're really fancy molecules that are produced by plants that are that have antiviral properties. And people are putting together formulations that are based on those. And I think that that black seed and the several, even just vitamin C and zinc, I mean, some basic things that people are recommending. And of course, sulfur containing amino acids like N-acetylcysteine and S-adenosyl methionine to take sulfur supplements. I think sulfur is really crucial because that's what's causing the immune cells to be weak. The immune system is impaired by glyphosate. I have a whole chapter in my book on glyphosate and the innate immune system. And I think it's being destroyed really by glyphosate. And so when people don't have a good, strong innate immune system, they have to draw in the adaptive system too much, and they end up with all these, the cytokine storm and all these reactive inflammatory response that injures the lungs and causes a cascade downward. It's a consequence of a broken innate immune system, which in part is due to the destruction of the mitochondria because of the deuterium problem. I really think the deuterium problem is central. I was so tickled when I found out that the flavoproteins played such a critical role in maintaining low deuterium, because I already knew that glyphosate disrupts the flavoproteins. And so that just made so much sense. I didn't hear about deuterium until December 2019. And I immediately jumped on it because it just seemed so perfect with respect to what glyphosate is doing to disrupt enzymes that are critically needed to maintain the low deuterium in the mitochondria.
Yeah, and then also real quick, I know I didn't want to take up too much time, but there's some.
Oh,
whoops!
Oh, something happened there, Alejandro. Somehow you
lost you.
Something's going on there, Alejandro, and actually, we were trying to get you back, but the truth is, we actually are up against the clock. So, sorry about.
We'll let that go.
Yeah, we tried to get him back, but sorry about that, Alejandro, and sorry to everyone else. I know there's a few other raised hands. It's time for us to move on to our next lecture. So thank you, everyone, and thank you, Stephanie. Thank you for coming.